Hey guys, so I actually yesterday, because I finished this up yesterday and I stopped it. I was having a couple of issues with the recording. It just stopped on me, and thankfully it stopped at a really good spot. But it was, yeah, I had issues, and so I decided to take a break. And so we're gonna pick it back up again today. So today we're gonna be looking at the rest of the elements of the adaptive immune response system, the immune response. And so I'm looking through your book right now, and we're basically at the end. We're going to be talking about B lymphocytes now. So I'm just going to make and I also got another pen, nice inky black pen. So that's hopefully going to help us make this final part of this amazing concept map, really. It's quite amazing. Okay, so B lymphocytes. First off, where do we find them? These B lymphocytes are pretty much found in various areas of the lymphatic system. So places like the spleen, the malt, right? The mucus-associated lymphoid tissue, lymph nodes, because remember, lymph nodes are going to be packed full of lymphocytes, including T lymphocytes. And then in addition, we're going to have some circulating the blood. So we're just going to write here, some circulate blood, which is really good for us. Okay, so pretty much the point of these B lymphocytes is to secrete antibodies, and we'll see how exactly they do so in just a second. But essentially what they do is they respond to specific epitopes, right? They respond to these specific epitopes which bind to the B cell receptors, which we'll talk about in just a second. They bind to the B cell receptors, and then once these B cell receptors are bound to the antigen or the epitope, the B cells basically start going through cellular division. They start multiplying and just making more and more of themselves. And so we have many of the same B cell that has the actual receptor, and therefore pretty much soon to be antibody against that particular epitope or antigen. And so at that point, once many B cells of the same B cells are made, we're going to secrete the immunoglobulin, the antibody, also known as the immunoglobulin, into the blood or the lymph. And then at that point, the immunoglobulin is going to start acting against the epitope that stimulated the B cell activation. Okay, so I'll talk about that in just a second. But so basically, to understand B cells, we have to understand BCR, or basically the B cell receptors, which are essentially pretty much the same as the T cell receptors. Okay, so each B cell has its own, has a unique set of unique copies of the BCRs. So this means that a BCR of one B cell is different from the BCR of the other B cell. So I'm going to write that down. I'm going to say one BCR of one B cell is different from BCR of other, that's O dashes other, of other B cells. So this means if one B cell can have thousands of the same BCR, right? And then another B cell is going to have thousands of another kind of BCR. And this is really important because this basically brings us a lot of different, a lot of just diversity and variability when it comes to B cells. And this is important because the more B cells we have that can enter and have their own specific BCRs, the more they're able to actually join and find complementarity with the epitope or the antigen. And so this is good because we want to make sure that all possible epitopes or antigens are accounted for in our system. So it's basically the same kind of concept with the TCR, except now that it's on the B cell. And we actually see a lot more variability in the BCR actually. So first off, what is a BCR composed of? These BCRs, remember, they're going to be attached to the B lymphocytes, but they basically look like the letter Y, right? And then this letter Y is then going to be attached via a transmembrane protein portion to the actual plasma membrane of the B cell. And the letter Y is really important. This is where we actually have the antigen binding sites. Okay, so this right here is the kind of the middle of the letter Y is the antigen binding site. You see one arm of the letter Y and the other arm of the letter Y. That's the antigen binding sites. So this is where the epitope actually binds to the particular the BCR. And so what it's made out of is it's actually made out of a heavy chain, actually 2 heavy chains and 2 light chains. 2 heavy chains and 2 light chains. And so they come together to look like that Y, and then the Y is actually put via a transmembrane protein slash anchor, you can call it, into the actual membrane of the B cell, okay? And so it's because of these heavy chains and light chains that we have so much variability. In fact, if you actually think about it in terms of genetics, I'm not going to get into too much detail, but essentially these, we have one of the heavy chains, we actually have 3 loci or loci of that actually, basically loci are just like locations of the genes that for 2 chromosomes that encode the various polypeptides that make up the heavy chain and the various polypeptides that make up the light chains. And so in total, we actually have 6 loci or 6 different positions together that can make segments that are DNA that are going to then be transcribed and translated into segments of the heavy chain or the light chain. Okay, so each loci in addition is actually like further divided up. It's further divided up into 3 different sections. You don't really need to know about the segments that much, but they're known as like the variable, the diversity, and the junction, right? So we have the variable heavy, the diversity heavy, and the junction heavy segments. We also have the variable light and the junction light segments as well. So we have all of these different kinds of segments that are encoded by these 3 different positions on these 2 different chromosomes. So 6 positions all in all. And so we have honestly about, I think I'm actually trying to remember this, I think it's about 348 different kind of genetic segments that are encoded, that encode these different like heavy chains and light chains. And the thing is that even though, so I'm going to actually write this down because I feel like numbers help. So 348 genetic segments, okay? 348 genetic segments. And then once these segments are made, they're going to be recombined. In fact, they're recombined with a specific enzyme known as, by recombine, just like come together to make combinations. It's a specific enzyme known as recombination activating gene protein, also known as RAG. So that RAG basically pulls these things together. It combines them in different various ways. And because of that, we can get a tremendous amount of variability. In fact, it totals up to about like more than 9 million different types of BCRs, just because the small little segments are just kind of combined together, right? Whatever, you know, segment they put, and then in whichever order, we can get more than 9 million different types of BCRs. In addition to that, in addition to various recombination, so I'm going to write various recombination, we can also have like point DNA mutations. We can also have deletions that occur, or even insertions. So various other kinds of mutations that can occur within the actual genetic segment. And this can result to, scientists approximate, about 10 to the power 23 different BCRs. Okay, so that's 10 to the power 23 different B lymphocytes that each have one of these types of BCRs that you have in your system. So we can basically have mount up an immune response against 10 to the power 23 different like antigens or epitopes. So it's absolutely mind-boggling and incredible how much variety we can get simply from these 6 different locations, like gene locations on our genome. And we can just get like a multitude of these B lymphocytes. And so they go ahead and like the B lymphocytes mature, right? Remember, they are made and they mature within the red bone marrow. And basically in the end, we can get, you know, a substantial amount of different kinds of B lymphocytes, and each one's going to have the ability to be find complementarity with an epitope, right? So we are basically set for life in this way. Because we found something that, you know, if something infects us, we're going to be able to mount an immune response towards it. And so basically when you do get an antigen, right? So I don't know where to write this. I'm just going to write it here. Okay. So when the BCRs do respond to an antigen, so responds to an antigen, it's going to be specific, you know, specific to the BCR of a specific B cell, right? At that point, the B cell is now activated, and in fact, we actually call it a plasma cell. Once the B cell is activated, it becomes a plasma cell. And what it means is that what it means when we say it's activated, it actually means that it's going to start dividing, so making more of itself, and then it's going to actually secrete. And it's going to secrete that part of the BCR, the initial Y part of the BCR, without that anchor, without the transmembrane protein anchor. And so in this way, it actually secretes antibodies, okay, also known as immunoglobulins. So it's going to secrete the antibodies into the blood or the lymph, and then the antibodies are going to react against the specific epitope, right? And the amazing thing is that once a bacteria or whatever microbe comes into your body, it's not going to just have one single epitope. It can have a multitude of different antigens. And basically this means that since say it has like 5 antigens, 5 different B cells can basically react and respond to each antigen, and each of those B cells are then going to then, activate themselves, divide themselves, become plasma cells, because they start secreting the corresponding complementary antibodies that, actually like the BCR component that had attached to the antigen. And so they're going to secrete that BCR component, take it away, and at that point, that's known as an antibody, right? It's basically the Y without the transmembrane portion. The antibody is going to be secreted, and then the antibody is going to be able to react against the specific antigen. So this is pretty amazing how it works. And so each antigen has an antibody that it's going to work against. Okay, so now we can pick up on, let's say, antibodies. So what actually are antibodies, and basically what their function is, right? So antibodies basically will bind, and they have a very strong, incredibly specific interaction. It's actually a non-covalent interaction with the epitope or the antigen. And so once they bind to that epitope, the antigen on the microbe, that's when we're going to have all sorts of different functions that they can actually carry out. So, okay, I'm trying to see where I can write this. I think I'm going to write it down here. So antibodies, okay, let's call it antibody function. Basically, they bind strongly and specifically to the epitope, and then they have various functions. Okay, and so these various functions, I'm going to kind of go like here, go across, right? And let's start out. So one of the first things that they do, and we kind of actually already talked about this a little bit, is activation of complement. And not just the complement, because once they activate the complement, it's going to trigger also things like inflammation. So they activate the complement and inflammation. In fact, the immunoglobulin mu, or IgM, is going to pretty much bind to specifically the complement 1 protein. This basically causes the complement cascade. It cleaves it and it activates it, so that we have the complement cascade. And then this can go ahead and actually trigger various inflammatory mediators as well. Another thing that happens is another immunoglobulin or another antibody known as IgE can actually bind to specific cells like eosinophils, and even mast cells, and this can also basically release more inflammatory chemicals. So I'm just going to write here, release inflammatory chemicals. Okay, so that's one way, is the complement and then the release of inflammatory chemicals. And we've talked about the complement already, right, in a past video. So that's how the complement, we're not going to go over how the complement works again. Another thing that they can do is actually opsonization, which is also another thing that we've kind of looked at already. Opsonization, so they can work as opsonyms. So they go around the antigen, say this is the antigen, and they will specifically, change its surface. So basically opsonizations, they act as opsonyms because they bind to the target, and this way they're going to change the surface of the antigen to make it more available for phagocytosis. So with opsonization, they just stimulate phagocytosis by changing the okay, by changing the surface of the antigen, so that like I like to think of it as the surface of the antigen is optimized optimized to be actually phagocytized. Okay. Another thing that they do is neutralization. This is kind of neat because we've been talking about how sometimes with various different diseases, if the infection is actually mainly caused by a toxin that's released by the bacteria, one way that we can actually treat it is by giving them an antibody that is specific to that toxin. And that's because some of our antibodies, they can actually neutralize. So they can bind to a toxin and neutralize it, or basically inhibit it from working. They usually is because they bind to a critical component of the toxin. So they neutralize a toxin. I feel like that's pretty self-explanatory. They can also neutralize like specific bacteria or viruses, just depending on where they bind. So say they bind to a particular area in the bacteria or a particular molecule in the virus that is necessary for adhesion and therefore for connection and interaction with host cells. If they bind to it, they're basically going to block the adhesion molecules stop them and inhibit them from inhibit them from carrying out their purpose, and so the bacteria can't go in or the virus can't go in. So neutralize, it's usually the bacteria viruses adhesion molecules. So that's another way that they can kind of neutralize the situation. What else? Oh, and then of course they can also work through it's known as antibody-dependent cell-mediated cytotoxicity. So that's kind of a mouthful. And so in fact, we actually don't call it that. We call it ADCC, antibody-dependent antibody-dependent cell-mediated cytotoxicity. So here, the antibody binds to the antigen, and it basically makes it it binds to antigen. So the antigen binding site will bind to the antigen, and the antibody stem, which we'll talk about in just a second. So that's basically if the antibody is like a Y, this is going to be the part that binds to the antigen, and this is the stem right here, okay? And the stem can bind to cells known as natural killers, and basically these natural killers, as their name suggests, these are specific lymphocytes that will kill and destroy the target cell. So since the antibody literally has brought the antigen to the natural killer, since half of the antibody can bind to the antigen, right? And the other half is going to bind to, I'm just going to call it like this is the natural killer. So now the antigen is right near the natural killer. The natural killer is going to, release certain kinds of, certain kinds of proteins known as perforin, which actually perforates and forms a hole. And then through it, it's going to it'll release another enzyme known as granzyme. And basically granzyme is going to go ahead and just kill, it kills the cell. It actually triggers apoptosis of the cell, triggers apoptosis. Okay. So basically in this way, the natural killer can go ahead and kill whatever, the cell, the cell that attached to the antibody via the antigen. So that's known as ADCC, or antibody-dependent cell-mediated cytotoxicity. And then the other thing that we have to consider is also agglutination. So I'm going to talk about it here, agglutination. And agglutination works because technically antibodies have two binding sites for antigens. Okay, so they have two binding sites for antigens. And so basically that means that they can attach to two epitopes at once. And so because of that, numerous antibodies can basically bunch up and attach to many different antigens together, and so they can aggregate the antigens. So we have two antigen binding sites, and so can bind to two epitopes, and so we can basically aggregate our antigens. And so with these aggregated antigens, they then form like an insoluble precipitate, okay, that can be easily filtered, or even phagocytized. Okay, so methods of antibody function. I know I kind of had to traverse the T helper cells a second, but antibody function methods, we have agglutination, ADCC, neutralization, opsonization, and then activation of the complement and inflammation. Okay, So now that we know how the antibodies actually work, we should talk about the antibody, what antibodies actually look like. Okay, so what antibodies actually look like. So actually, I think I have it here on the screen, on my PowerPoint, so you can see. So antibodies have a heavy chain, 2 heavy chains, and 2 light chains. And as you can see, there's on one arm there's an antigen binding site, and then in the other arm another antigen binding site. This is what I meant by 2 antigen binding sites, which allows for agglutination. The area that is variable, and that basically is going to, is specific to whatever kind of epitope. This is known as the FA, like the FAB, I call it FAB, the FAB region. Okay, this is the fragment antigen binding region. And then, of course, we have the hinge, and then we have the stem. The stem is also known as the FC. And basically, there are 5, and this is basically composed predominantly, only of the heavy chain. And there's going to be 5 types of stems. And these 5 types of stems, mu, G, A, E, and D, are going to constitute the 5 classes of antibodies. And so these 5, these FC regions of each class is basically in a person's body are going to be identical. So all the IG, so if we have the FCG, right, FCG, we have IgG. That's the particular immunoglobulin that we have, the particular antibody that we have. And that means that all of your IgGs are going to have the same FC region. Okay, so they're basically, this is what an antibody looks like. They're just secreted BCR. So if we go back, how do I stop share? If you guys remember here, right? These are the BCRs. Do you remember we have technically the immunoglobulin here. So we have the light and heavy chains, and then they have a transmembrane protein. Basically, the majority of your antibodies are secreted. And so right here, antibody structure. We have the variable region, which is also known as FAB, and then we have the stem, right, which is known as the FC region. And the stem, we have 5 different types. So 5 types, which constitute the 5 classes of immunoglobulins, and we'll see what they all look like as well. Okay, so basically an antibody is a free, for the most part, immunoglobulin that is not attached to the membrane. So free immunoglobulin that is not attached to the membrane, and they of course have all these things. So they're similar to BCRs, but they actually lack the transmembrane proteins, and hence the fact that they're not actually attached to the membrane. And so they have that Y, right, that Y structure with like a heavy and like the variable chain right here that's made out of heavy and light chain, which is going to constitute one binding site, and then the heavy and light chain on the other side, which is going to constitute the other binding site. So we have 2 antigen binding sites, which relates it back to them being able to go through agglutination. Okay, so what are the different kinds of antibodies? Well, the classes basically are, we have 5 types. We have 5 classes. And the classes that end up going to be in, like, whichever one gets actually involved in the immune response, this depends on a variety of factors. It depends on, of course, the antigen that is being presented, so the foreign antigen that's being presented in the first place. It's also going to depend on which kind of function of the antibody we need to actually work, and of course also the portal of entry. And so we'll see some are actually secreted, like some are actually secreted, some stay in, some are long lasting, all sorts of things. So basically we have, if you remember, the plasma cells, which are going to be your activated B cells. The plasma cells, they begin by, attaching the variable region. Like they start basically like releasing these antibodies. And these antibodies are always going to begin by making the immunoglobulin mu. So IgM or IgMu, okay? That's going to be that first initial antibody that is made. So the FC region is the mu region right here. So that's the stem region. So they're always going to start by just adding a variable region. The variable region, remember, is a part that binds to the antibody to the M stem, the mu stem. It's actually not M, I keep saying M, but it's the mu, to the mu stem. So these are going to be your initial ones. So initial They're secreted in the initial stages of the immune response, right? They're actually quite bulky. They're quite big. I think I believe I have them on my PowerPoints, but I'm not 100% sure. Like the different types of immunoglobulin. Oh, here you can see like the different functions of antibodies. There we go. So you see IgMu, right? You see how big it is. So this is the first kind of immunoglobulin that is actually made. And then we go through something known as a class switch. So class switching is when the plasma cell, remember that the activated B cell, it goes ahead and it combines the variable region, like the variable regions of, two different of that they have towards that particular antigen, two different stems or two different FC regions. And so that's when we get IgG, IgA, IgE, and sometimes even occasionally IgD, which is kind of uncommon. Okay, so back to IgMu. That's going to be secreted as a very bulky one. It's actually a pentamer, so there's like 5 subunits to it. And basically, when it does get secreted in those initial stages of the immune response, we're going to have things like complement gets activated. We also have, there's complement activation, which of course then leads to triggering of the inflammation as well. They're also really good at agglutination, right? Especially that pentamer structure really helps with that. And then of course we have neutralization as well, okay? So that's your first one. After it, we're gonna have class switching. So I'm gonna write here class switching. That's when they put the variable on different kinds of stems. And that's when we can get IgG, right? This is actually your most common one. Most common is actually also the longest lasting, so long lasting. Most common and the longest lasting type of immunoglobulin, and so, or antibody. After that, it basically is involved in pretty much everything. It's involved in complement, it's involved in opsonization, it's involved in the antibody dependent cell mediated cytotoxicity, ADCC, and it's also involved in neutralization. Another thing about this is it's a lot smaller than IgMu, and so because of this, it can actually leak through the blood vessels, and it can go into this extracellular kind of space. So it can really travel into different areas and really like attach to the pathogen and neutralize it if it has to. And because it can leak through and it's really small, it can also, this is the antibody that can cross the placenta and therefore kind of protect, right, defend the fetus, the baby. So that's really important. Then we have IgA, which I'm going to put right here. This web is a bit too, like I think my page is a bit too small. Sorry. IgA. This is also known as secretory like immunoglobulin A. This is because it is secreted. And it's made out of A because it's made out of alpha heavy chains. So this is associated with body secretions. So anything like secretions from mammary glands. So as you can imagine, this is going to be found in the milk. So this is going to protect the newborn baby. It can also be found in things like tear ducts or, like the mucous membrane as well. So this is going to be secreted. It works by agglutination and neutralization. Okay. We also have IgE, which is made out of epsilon heavy chains. This is usually found quite low in concentration actually in the serum, and it's more of a signal molecule where it will bind to specific receptors on things like eosinophil cells, or even I believe even mast cells and basophil cells. So with eosinophil cells, it's going to start like it's binding, it binding to these kind of cells. It's going to trigger a release of cell damaging molecules, especially if you guys remember, these eosinophil cells work very well against parasitic worms, and so that's where the cell damage is actually going to happen. They can also trigger things like mast cells or basophil cells to release more inflammatory chemicals. So basophils and mast cells, so they can release inflammatory chemicals. So chemicals like histamine, for example. And then we have IgD, or delta. It's made out of delta heavy chains. So this is the only one that is not secreted, or basically that is not, it's membrane bound. So this is your BCR that stays attached to the membrane, which I know is very weird because all of these aren't attached to it because the plasma cell activates and makes, like spits out antibodies. IgD is the only one that stays like membrane bound. So it's essentially like your BCR. So yeah, membrane bound antigen receptors on the on your B cells. So a lot of times we can see them in the initial phases, but frankly, like, we don't really know too much of this, especially since not every single mammal has it. And so we don't really understand the function of this as well. So yeah, what else does your book talk about? Oh, clonal deletion. Okay, so clonal deletion, I should have talked about it when we were talking about BCR, but basically when all these BCRs are made, because remember we have numerous combinations of BCR, your B cells have to mature, right? And so we have to make sure that none of the BCRs are made against self, or none of the BCRs are non-functional. And so of course, if you remember the B cells, this happens in the bone marrow, right? Any kind of self-reactive B cell is actually going to be changed. So unlike the TCRs, where any kind of, like, when they mature in the thymus, they're going to go through apoptosis so that we get rid of any self-reactive receptor, or any non-functional receptor, we get rid of it. With the BCRs, they actually, instead of like going through apoptosis, they simply change so that they can hopefully become, a good reactive B cell that is not self-reactive or that is not inactive. So we just have maturation. I'm just going to write maturation to change self-reactive BCRs and non-functional BCRs. Okay, and so we change them. They don't actually go through apoptosis, even though it's still called clonal deletion, right? They just change. So once we have these tolerant BCRs on the the BCRs on the B cells, they leave and they travel to the spleen, right? Here they actually go further maturation, and then once they get to the spleen, they can then circulate in the blood and lymph, right? And so we're going to have both our T cells and our B cells circulating in the blood and lymph so that they can basically like scan antigens together, and they can communicate with each other via cytokines. So these cytokines are the next part of, these go here, but I need to connect it from here because it's another element of the adaptive immune response. And actually, it's not just in the immune response. You saw it also in the innate immune response as well. So how am I going to do this? Dang it. Just shamelessly go through all of this. So we have, and I don't know why I made a little arrow. I haven't done arrows. Oops. Oh, I've done an arrow here. Okay, anyways. So just cytokines. Hold on. Okay, cytokines. And this is the last part, I think, and then I'll end this video. So cytokines, these are basically intracellular messages. These are regulatory proteins that are soluble, that are secreted by like your, various leukocytes, that go ahead and like regulate things. And so basically because of that, we get like the cytokine network, right? Because all of these regulatory proteins kind of like add, like making things work in your immune response. So we have the cytokine network because there's such complexity and, just variety in the cytokines, and some of them are also quite identical as well. So like the same ones can, allow for the same thing to occur. So that's where we get the cytokine network. So it's just a complex web essentially of all these signals that are traveling between cells of the immune system. So I'm just going to write here complex web. So it's just a bunch of signals between cells of the immune system. So they're actually released by leukocytes, right? So they're secreted by all these leukocytes, and they are regulatory signals. So, okay, there's a few different kinds. So I'm just going to write here types. And so let's move on. So the first one, interleukins. Actually, I shouldn't have written it all. ILs, interleukins. These are going to be, they're secreted among, leukocytes, so nothing new here. You also have things like growth factors, for example. I'm just going to call them GFs. Growth factors. So growth factors allow for cells to grow. So they stimulate specifically, and they grow more cells of that make leukocytes. So they actually stimulate leukocyte stem cells. Stimulate leukocyte stem cells. So basically, we're replenishing our leukocytes in our body. So yay, we have enough immune system cells in our body to divide. We stimulate them to grow more and make more of themselves. Another kind that we have, and actually, we already talked about this one a little bit, interferons. Remember, these guys are the ones that work against viruses. They inhibit, so that's right, inhibit viruses. They don't like viral infections. And we actually talked about specifically the gamma interferon, right? Gamma interferon. And so this is, yes, to see how it can kind of it can what's the word I'm looking for? Overlap. Yeah, it can overlap with the innate system as well. So interferon, like gamma interferon, that can basically also act as a cytokine as well. And so it actually goes ahead and activates phagocytosis. It's activated with the interferon. And then we also have the tumor necrosis factor, which, as you can imagine, kills tumors, right? But it doesn't just do that, it kills tumor cells, hence the name. It also goes ahead and, since cytokines are regulatory signals, it also goes ahead and regulates the immune response and even inflammation. So part of the innate as well. These are going to be secreted by things like macrophages, and even T cells as well can also secrete the tumor necrosis factor. And then I think finally we have chemokines. Chemokines, which are actually chemotactic. The word chemokines are chemotactic. Tactic, like taxi, this means movement. So this allows for the movement. It signals, because remember cytokines are signals. So it signals the various leukocytes to move. Signals leukocytes to move. So they can move to an area of inflammation or an area of where the infection is happening or wherever in the body. So that's basically the chemokines. So those are cytokines, which are really important to remember, because this is how the lymphocytes and leukocytes, they communicate with each other, and they help each other scan the antigens in the blood and the lymph, etc. So that is it for the elements. Oh, goodness me, look at that. Elements of the adaptive immune response. Like I said, I'm going to take a picture of this and upload it. And then I believe we're almost done. We're just going to talk really quickly about cell-mediated immune responses and antibody immune responses as well. All right.